Hi. So, in previous session, we have initiated the innate immunity. We have discussed why innate immunity is needed and how the innate immunity is playing a crucial role in our everyday defense, uh, everyday defense against array of microbes, which include microbial pathogen as well. And then we have discussed about various component of innate immunity. We have looked at the physical barrier, which is also known as mechanical or anatomical barrier. We have discussed chemical and biochemical barriers. Okay, uh, and we have discussed the. I just introduced this chemical and biochemical barrier and microbial or microbiological barriers. Okay. So, in previous session I have discussed in great length about skin, which is a mechanical or anatomical barrier. Today I will take you further and I will discuss in more detail about the mucosal surfaces. So, this is also anatomical barrier. Okay. And I will also talk about some of antimicrobial uh, enzyme or protein or peptides, how it works. And I will also discuss maybe in this session or next session about the microbiological barrier. Okay. Okay, let's begin with mucosal surfaces. And mucosal surfaces is quite huge, right? If you remember, I have discussed these surfaces, and it's a huge space, it's a huge area which constitute about 400 square meter. Okay. So, these mucosal surfaces are present in the gut, in respiratory tract and urogenital tracts. Okay. And gut is one of biggest place. Okay. So, what is these mucosal surfaces? This is a line all, this is a lining all body cavity which opens to the outside environment. Okay. So, the lining which is present inside. Okay. And this lining basically opens outside. Outside means if you look at oral cavity, if you take urogenital tract or respiratory tract, it opens outside the environment. Okay. Unlike uh, skin, it's the epithelial cell which is present in this area. Basically, this is a living. Okay. And this is a living, and there is a few more things are associated, that is just beneath these layers there is a some loose lymphoid tissues are there. Okay. And you may remember that in order to transport the antigen in the to the lymphoid tissue, there is some specialized cells are there which we call it as a M cell. I have discussed during discussion about mucosal associated lymphoid tissue, okay, or mucus associated lymphoid tissue. Okay. Epithelial cells in this mucosal area, the epithelial cells are again very tightly packed like a skin. Okay. And this tight packing of the cell is basically prevent the entry of any microbe. I want to say microbe, it could be a pathogenic or non-pathogenic. Okay. But since it is a only single cell thick, it's a only single layer of a cell, so several pathogen they can breach or they can break and enter in the in deep tissues. Okay. So there are several pathogens which is infecting through oral route. Okay. Those pathogens develop some mechanism, and some of these pathogens, they have some anchor like protein which hooks up with these epithelial layer, and then they gain access uh, inside the tissue, and then they can infect. Okay. So most of these microbe, especially the pathogenic microbe which cause some disease, they have this unique feature. Okay, they have some or other way by which they can hook up to these cells, and then they can enter. Okay, but on another hand, the host defense side, if you see, these cells are quite quickly removed. Okay, and whatever pathogen which is adhered, okay, to these surfaces, they are thrown out. Okay. So I'm just trying to explain there is a evolution from both sides, the pathogen side as well as from the microbe side. So this is a kind of battle. Okay. And one more interesting thing is that these microbial pathogen, they are uh, kind of their ability to cause disease is basically depend on the dosage. Okay. So if the that's why we always emphasize the number of bacteria. Okay, or initial dose of infection. Okay. So that also determines whether it will infect or cause disease or not. Although if the dose is very low, they can infect, but they may not cause the disease, because if the dose is very low, maybe our initially the defense system will wash off all these microbial pathogen, and then we will be free from the disease. But if the dose is too high, then there is a high probability that in some case these microbes or these microbial pathogen gain access in the tissues, and then that will cause the disease. Okay. So this is all there are so many parameters, try to understand, it's not only one thing. Okay. So as I have already told you that these layers are continuously shedding off. Okay, and whatever microbes are attached, they will be thrown out. Okay. In addition, there are some cells which produce a lot of amount of mucus. Okay, I will show you in subsequent slide. Besides this, there is some these cells or the layer of these cells. They basically produce a variety of antimicrobial enzyme, such as lysozyme, which is very well characterized, and there are some antimicrobial peptides. I will discuss in this session or maybe next session uh, about few antimicrobial peptide and how they work. Okay. So all these things basically protect these mucosal surfaces from invasion of microbial microbes or microbial pathogen. As I told you, there is a continuous production of mucus. Okay, and this mucus, if it's very interesting that if you know that we produce about a liter of mucus every day, and we swallow that much amount of mucus. Okay, so this is a very good defense mechanism. You are protecting against this variety of microbe as well as microbial pathogen by, because our body is, or our epithelial layer, there are some cells which keep on producing this mucus which has a washing action, and this washing action prevent from lot of infection. Okay. But as I told you, these microbial pathogen, they have some unique system by which they can hook up and then they can gain access. It's true for bacteria as well as fungi. Okay, sorry, bacteria and viruses. Fungal infection in gut is not so common. It's very rare. I never heard. So it's a not fungal infection, mainly bacterial and viral infection. Okay. Now I will take you to the mucosal surface lung. Let's look at the lung. Here you can see that lung is a quite big area. Okay, and there is a this is a lining of lung epithelia. Okay, and here you can see there is a one specialized cell which keep on producing mucus. Okay, which we call it as a goblet cells, and this secrete mucus, and this mucus basically helps in trapping the microbe and ejecting the microbes, and the production of mucus along with the ciliary movement of this epithelial lining. They have a cilia, and these cilia is keep on moving. Okay, so this the mucus coupled with ciliary movement of these epithelial cell basically throw out the microbes. Okay, and by this way we can protect the host from infection. Okay, now I will show you the mucosal surfaces in gut, which is a quite huge area. Okay, here also you can see there is a goblet cells, okay, which is producing mucus. There is a Paneth cell, and these Paneth cells basically produce variety of antimicrobial peptides or protein that like alpha defensin, regal. Okay, and there is a crypt, and this mucus is keep on moving, so the microbe cannot attach. Okay, and if it will be attached, then yeah, it will be taken to the lymphoid tissue, which is just present beneath this thing. You probably remember there is a Peyer's patch, lamina propria. Okay, over there is a lymphoid tissue, and this will be taken care by that. Okay, so this is the mucosal surfaces are also protected by variety of microbes. Okay, I told you in previous session there is a commensal bacteria, and there are some bacteria which has a mutualism or mutual relation, means both are getting benefit. Okay, so those microbes are also present, and due to those microbe, the infection by pathogenic microbe is relatively less. Okay, so I will discuss in microbiological barrier. Okay, so this is about the mucosal surfaces, and now I will talk about some or other antimicrobial protein, enzyme or peptide. So first is the lysozyme. So here you can see the structure of gram positive and gram negative bacteria. Here you can see there is a, in gram positive bacteria there is a very thick layer of peptidoglycan. Okay, and this peptidoglycan is very thin in case of gram negative bacteria. Okay, and this gram negative bacteria has one more additional structure which we call it as a outer membrane. Here you can see there is a outer membrane. Okay, and this outer membrane contains your the most immunopotent molecule, which we call it as a lipopolysaccharide. Okay, so this lipopolysaccharide is present in gram negative bacteria, as you might know in previous classes, that the gram negative bacteria has an outer membrane, and this outer membrane contains the lipopolysaccharide. So what lysozyme is doing here, you can see this is a very nice cartoon, and lysozyme basically cleave this peptidoglycan and break or disrupt this peptidoglycan layer and open the membrane part of bacteria to the environment, and then there is several ways by which this can be disrupted. Okay, once the wall will be disrupted, then bacteria will be no more live. Okay, so this is about the lysozyme. In next session I will talk about some more antimicrobial peptide protein, and I will also discuss about the microbiological barriers. Okay, thank you.